the biological fixation of nitrogen. There are certain living bacteria which is responsible for the fixation of nitrogen in the soil by two ways. One is the free living bacteria and second one is the symbiotic bacteria. Free living bacteria, they are again classified into two types. One is the aerobic bacteria and the anaerobic bacteria. The example of the aerobic bacteria are Azotobacter and Beijernickia. These are the two aerobic bacteria which is responsible for the fixation of nitrogen in the soil, and they are available in the free living. The second one is anaerobic bacteria. The example is Rhodospirillum. Apart from this, certain Bacillus bacteria and in the blue-green algae means cyanobacterium. Blue-green algae, or it is called the cyanobacteria, are Nostoc and Anabaena. They are also the free living bacteria which is responsible for the nitrogen fixation in the soil. So, nitrogen fixation in the soils by two ways, free living bacteria and the symbiotic way. In the free living way, there are again two types, aerobic and the anaerobic. This is aerobic and this is anaerobic. The example of the aerobic bacteria are the Azotobacter and Beijernickia, and the anaerobic bacteria are the Rhodospirillum. And the other bacteria, that is Bacillus, and the example of the cyanobacteria are Nostoc and Anabaena. They fix the nitrogen in the soil. So this is all about the free living bacteria. Now next is the symbiotic bacteria. So in the symbiotic bacteria, there are again two ways. One is the symbiotic with the legume plant. We all know that legume plant means the plant which possess or produce the pulses. They are known as the legume plant. And the second one is the non-legume plant. Example of the legume plant are the alfalfa, sweet clover. First is alfalfa, second sweet clover, sweet pea plant, lentils, garden pea, broad beans, clover bean, etc. are the legume plant. And this all legume plant root possess the bacteria known as the Rhizobium bacteria. That Rhizobium bacteria live symbiotically in the root of this legume plant, and they help in the fixation of nitrogen in the soil. Why this association of legume plant, root of the legume plant and the Rhizobium bacteria called the symbiotic association? Because this Rhizobium bacteria provide the nitrogen to plant. It provide the nitrogen to plant. Plant synthesize the food, and that food is utilized by the bacteria. Means the bacteria provide nitrogen to plant and plant provide the habitat as well as the nutrition to bacteria. That's why this relationship is known as the symbiotic relationship. The another example is the non-legume plant. Example is the Alnus. Alnus is the non-legume plant, and the name of bacteria is the Frankia. So that Frankia bacteria is live symbiotically with the Alnus plant. Okay, so there are two bacteria. One is Rhizobium and second one is the Frankia bacteria. They live symbiotically with the legume plant and the non-legume plant respectively, and they help in the fixation of the nitrogen in the soil. Now how the legume plant, root of legume plant and the Rhizobium bacteria are associated. So the Rhizobium bacteria forms the root nodules. They form the root nodules in the root of legume plant. Okay, so how this bacteria, Rhizobium bacteria, form the root nodules? The next point is the root nodule formation. Okay, so this is the root, tap root. It's a tap root of legume plant, and this is the root hair. The Rhizobium bacteria get multiply and they arrange, surround the root of or the root hair of the legume plant, and this is called the rhizosphere. This is called rhizosphere. Now this bacteria get multiply and they arrange surrounding the root hair as well as the epidermal cell of the root. Now in the next step, the root hair get curled. Root hair get curled and the bacteria enter or invaded. Bacteria enter or invaded in the root hair and they migrated up to the cortex part of the root. Cortex cell. Okay, we know that when we take the section of the root, the outermost layer is called the epidermis, and below the epidermis there is a multiple layer of the cortex. So this is the root hair. So bacteria can penetrate in the root hair and then they travel and multiply in the cortex area, where the bacteria get multiply and they form the tumor-like structure or they form the nodule-like structure. It's called the root nodules. Okay. Again, how the root nodules get formed? The root nodules formed with the help of Rhizobium bacteria. First, the Rhizobium bacteria is grow or multiply surrounding the root of the legume plant, and this group of bacteria is known as the rhizosphere. Now in the second step, the bacteria is surround the root hair and the epidermal cell of the root. Then in the third step, the root hair get curved and the bacteria get penetrated or invaded in the root hair and they migrated up to the cortex part, where it get multiply and they form the tumor-like structure that is known as the root nodules. Now, once the root nodules get formed, this bacteria release into the cell. Now the bacteria get released into the cell and now this cell is known as the nitrogen fixing cell. And this nitrogen fixing cell means, for example, here the bacteria is released and released into the cell. Bacteria released into the cell. Now this cell is known as the nitrogen fixing cell. And this nitrogen fixing cell possesses the two main components. One is the nitrogenase enzyme and second one is the leg hemoglobin. Leg hemoglobin is the leguminous hemoglobin. Now this nitrogenase enzyme possesses iron and the iron molybdenum component. And this nitrogenase enzyme is help in the fixation of nitrogen. This nitrogenase enzyme is sensitive to molecular O2, means it is sensitive to molecular O2, means in presence of oxygen, this nitrogenase enzymes cannot able to perform their functions. It required the anaerobic condition. So who provide the anaerobic condition for the activation of the nitrogenase enzyme? So the leg hemoglobin, actually in our body hemoglobin is there. This hemoglobin binds with the oxygen. Similarly, the leg hemoglobin, it's a scavenger. Leg hemoglobin is the scavenger of O2. It will scavenge the O2 and it provides the anaerobic condition for the enzymes. Okay. So, the Rhizobium bacteria, actually they are aerobic bacteria. Rhizobium bacteria are the aerobic bacteria. They are aerobic bacteria, but during the nitrogen fixation, this bacteria get anaerobic. Clear? Now, this nitrogen, how the nitrogen get fixed? So, this is the nitrogenase enzyme. Bind with the two molecules of two atoms of nitrogen, and as we discussed earlier, the nitrogen are combined with the help of the three strongly covalent bond. Now with the addition of the hydrogen atom, the one bond get break and it bind with the two molecules of or two atom of hydrogen and it form the 2NH. Again the hydrogen will be added, one more bond get break and it form the NHH. Again the two hydrogen will be added and it form the NH3. Okay, and at the end it forms the 2NH3 and the enzymes are released. The whole process required the 16 ATP. Here the equation is nitrogen plus 8 electron plus 8H plus and 16 ATP form 2NH3, 2 hydrogen atom, 16 ADP and 16 inorganic phosphate. So with this equation we know that the formation of the 2 ammonia, it required the 16 ATP. So if the formation of the 1 ammonia, it required how many ATP? It required the 8 ATP. So this is all about the biological nitrogen fixation with the help of the various living organisms. Clear? Okay. Let us discuss again that biological nitrogen fixation, free living bacteria and the symbiotic bacteria. In the free living bacteria, aerobic, Azotobacter and the Beijernickia, and anaerobic, it required the Rhodospirillum. Other bacteria are Bacillus and the blue-green algae, that is Nostoc and Anabaena. Okay. Now, symbiotic, that is legume plant and the non-legume plant. Okay. Legume plant is alfalfa, sweet clover, sweet pea, lentils, ground pea, broad beans, clover bean, etc. Where the rhizobium are the bacteria fix the nitrogen. Non-legume plant Alnus and the bacteria is Frankia. Root nodule formation, the first step rhizosphere is formed. Second step the bacteria gets around the root hair and the epidermal cell. Third step root hair get curled and the bacteria enter into the root hair and it form the infection thread. Where fourth step the infection thread get release the bacteria get multiply and it form the nodules or the tumor like structure. This bacteria release into the cell and the cell is known as the nitrogen fixing cell. This nitrogen fixing cell has the two things, one is nitrogenase enzyme and second one is the legume-hemoglobin. This nitrogenase enzyme contain the iron and molybdenum and it is sensitive to molecular O2. It requires the anaerobic condition for the performance. Who provide the anaerobic condition? Leg hemoglobin. The leg hemoglobin is scavenge the O2 and it provides the anaerobic condition. So the rhizobium, actually they are aerobic bacteria, but in presence but in the fixation of nitrogen, it acts as an anaerobic bacteria. And for formation of the 1 ammonia, it required the 8 ATP and the formation of 2 ammonia, it required the 16 ATP. Clear? So this is all about the biological fixation of nitrogen. Now, next point is the fat of ammonia. Means when the plant absorb the ammonia, okay, this ammonia is converted into amino acid and it synthesizes the protein.